Yes, ma'am. I've been instructed not to eat the microphone. Okay. So this is an aspirational title. This is what we would like to try to do, is identify pathogenic B cells in type 1 diabetes. So before I get too far, I always like to acknowledge the people who have done the work. And there's a huge team involved with this, as you can imagine. So they're the people in my lab, Weng Zhao, Jamie, Fang, Zuming, Jake, and Aaron, who have all worked very hard on this. And we have some very nice new collaborators from Stony Brook who have been helping us with some of the antibody modeling. There's also the Human Pancreas Analysis Program, which I'm going to tell you about a little bit. That is a huge effort to bank and characterize tissues from organ donors. And then there's the Penn Autoimmunity Center of Excellence, our collaborators there, and also the Human Immunology Core at Penn. Oh, and last but not least, actually, I should acknowledge the donors, their families, and of course our sources of funding. All right, so I'd like to talk to you today about the identification of potentially pathogenic B cells in type 1 diabetes. And I always like to start talks with telling you what I'm going to say, then I'm going to try to say it, and then concluding with what I have said. So this is sort of the rule of threes, right? So T1D features is what we're going to talk about first. Why do we even want to look at B cells? And then talk a little bit about how do we approach homing in on potentially pathogenic cells. And I'm going to tell you about what I like to call bottom-up and top-down approaches. I'll describe the Human Pancreas Analysis Program to you a little bit. And then we'll dig into some data sets that look at some features of cells that we think may have more interest in T1D pathogenesis potentially. And last but not least, I'll describe a little bit of new data on antibody modeling and analysis of some very recently generated monoclonals, and then how might all of this be useful. So we'll zoom back out at the end. Okay, so really quickly, I know many people know this, but just a brief primer on type 1 diabetes. So of course, this is an autoimmune disease that's characterized by the loss of beta cells in the islets of Langerhans in the pancreas. The result of this is while you have a T cell infiltrate, usually CD8 T cells that are destroying these cells in the islets, the beta cells get destroyed. These are, of course, the cells that produce insulin. So then you wind up with a lifelong dependence on exogenous insulin, glucose monitoring. You have impaired glucose metabolism and a whole slew of very unpleasant medical complications. Now, why would we want to study B cells in a T cell-mediated disease? So actually, Andreas Losius from a few days ago mentioned this in his talk on MS. So type 1 diabetes and other classical T cell-mediated diseases do have roles for B cells. And one of the main roles in the case of type 1 diabetes is antigen presentation. So B cells in T1D present antigens to pathogenic T cells, and if you knock out antigen presentation function in mouse models, such as the NOD mouse, you can very substantially alter the likelihood of disease development. And those were studies that were done now more than 20 years ago by Huma Norkazem and Ali Naji. We showed many years ago that people with type 1 diabetes have altered B cell subsets in their circulation, and that they also, some of them have defects in receptor editing of their antibody genes, which also point potentially to B cell involvement in the disease. There's, of course, also a literature on autoantibodies that are produced and are predictive of disease, which I'll show you in a minute. And perhaps the most convincing demonstration in humans is that if you deplete individuals who are in the honeymoon period of type 1 diabetes, which is the period before disease development, but after sort of high risk of getting disease, such as the presence of 2 or more autoantibodies, that if you give those patients an anti-CD20 therapy, you can prolong the period of time before they require insulin. So, but they do eventually get disease. Also, I think it's important to mention that B-cell targeted therapies in humans are generally very well tolerated compared to T-cell targeted therapies. And so if there's a way to disrupt the connection between T cells and B cells, even in a T-cell mediated disease, it's useful, right? So studying how B cells can do this is ultimately part of our aim. So the short answer to this question is B cells help T cells. And so they can be involved, and trying to define them and potentially disrupt them could be a means of improved diagnosis, monitoring, and therapy for T1D. So T1D also has, and that's type 1 diabetes, which is, I'm lapsing into the abbreviated form of the disease description. The T1D has major autoantibodies that are produced in individuals, and sometimes these are produced months or years before disease development. There are 4 classical specificities that have been described. So there's IA2, zinc transporter 8, insulin, and GAD65. And in this paper from already now over 10 years ago, large-scale study in humans looking at the number of antibodies against these antigens, and then looking at the risk of disease development, you can see that with 2 or more autoantibodies, your risk, if you follow these individuals out long enough, is over 95% actually, if you have 2 or more autoantibodies. In contrast, if you have one autoantibody, then your risk is not negligible, but it's way lower. So there is something that happens between the formation of 1 autoantibody and 2 that we do not really understand. Okay, so our goal is to really try to find pathogenic B cells in type 1 diabetes. And looking in the blood is a difficult thing to do for this because the blood is like an ocean and there are many different B cells in the blood. And usually only a very small fraction of those are going to be relevant to disease. So our strategy has been to leverage the Human Pancreas Analysis Program, which has type 1 diabetes donors, also donors who are autoantibody positive, as well as healthy control donors, and then analyze B cells in these donors using 2 different but complementary approaches that I like to call bottom-up and top-down. So bottom-up is, or guilt by association, if you will, is that you analyze global features of the immune repertoire in the tissues of these organ donors, and you look for differences between type 1 diabetes or autoantibody positive donors and control donors. You define the anatomic location of B cells, you look at features of the repertoire such as somatic hypermutation or clonal expansion, and then you look for clones of B cells potentially that are enriched in either particular tissues such as pancreatic lymph node or particular B cell subsets. So that's bottom up. It's kind of agnostic to antigen, right? Top down, on the other hand, is to sort on autoantigen-binding B cells and then analyze their repertoires, look for evidence of selection, and then also clone and express some of these antibodies that these cells produce, confirm their autoreactivity, and potentially also evaluate multi-reactivity. So, okay. So I mentioned that this all relies on the Human Pancreas Analysis Program, which is an NIH-funded program that's carried out at the University of Pennsylvania, University of Florida, and Vanderbilt. This is an amazing resource, actually. They've collected over 170 organ donors, and you can see the data are shown in this database called PanCDB. I'm not sure this is working. Yep, there we go. You can access these data sets online, and for each donor you have lots of different data that you can dive into, which is very nice for deep characterization profiling of the donors. We have lots of different donor types, controls, T1D, there's also T2D in there. And importantly, we have autoantibody positive donors who do not yet have metabolic evidence of impaired glucose homeostasis, but do have 1 or more of the 4 classical T1D associated autoantibodies. So these autoantibody positive donors can be used as an intermediate state in disease development from being healthy to having full-blown disease. Okay, so now let's start with the first experiment of what's bottom up, okay? So we've been generating bulk sequencing libraries from spleen from these Human Pancreas Analysis Program donors, and here we're taking 14 different donors per group. So we have T1D, autoantibody positive and healthy control, and you can see some of the features of the groups. They're not super well controlled because they're low numbers, but you can see the age distribution there, the sex, and importantly, the autoantibody counts. So you can see that the dark wedges here is 4 autoantibody, 3, 2, 1, and 0. In the autoantibody positive, most of the donors here are single positive, but some are double and triple. And of course, the healthy controls don't have any autoantibodies. Okay, so we're going to sequence antibody heavy chain rearrangements initially and just look at the landscape of the repertoire and ask some very basic questions like, do T1D patients have, or organ donors have larger B cell clones? Do they have any shifts in their V gene usage? Things like that. All right, so when you first start to look at data like this, the first thing we look at is just kind of the metadata. And the clone counts here are not evenly sampled. So that we need to fix. So we subsampled donors down to the level of 20,000 clones per donor. We define clones in our bulk sequencing data as having the same V, J, CDR3 length, 85% amino acid identity in CDR3. And then we further group some of these clones together based on CDR3 nucleotide similarity. Okay, so once you do that, then you can start to look at whether you have large expanded clones in T1D compared to control. So one way to do this is to just take a cut off the highest, most frequent clones using something called the D20 index, which is the fraction of rearrangements in your sequencing library, sequence copies, that are occupied by the sum of the top 20 ranked clones. And in a diverse pool, in a diverse repertoire, this number is usually pretty low. And here it is generally also pretty low. It's a little higher than what you would see in the peripheral blood, but it's still pretty low. So in this plot, each dot is a donor. You see that if you have T1D, which is in red, this D20 value is anywhere from 1.5% to 0.5%. The autoantibody, there's one outlier there. And then healthy controls are a little bit more tightly clustered. But again, not significantly different looking if you just do averages like this of the D20 index. If you want to cut down a little further on the distribution of clone sizes, again, here red is T1D, purple is autoantibody, and blue is control. They really look very similar, right? So it's not like T1D individuals, at least if you just grab spleen in bulk, have some really large clones to look at. Okay, what about V gene usage? So this is one of these very deceptive waterfall plots where you can show really tiny differences, right? So that they're not very big. So if you look at the difference in the frequency of V gene usage, and now we're kind of shifting over into red is going to be T1D plus autoantibody versus blue being just control. You can see there's a few V's that are overrepresented, but the very first is our friend V438-2, which years ago Corey warned me about this V gene. It's kind of a difficult one because there is actually a high rate of deletion of this V gene in the human population. So what we think is going on here is, and it's still interesting, but it's a different interpretation, is that individuals who have this gene are more common in our donors that have T1D or are autoantibody positive. So that's an allelic difference, not necessarily a frequency difference, because if you take, and I'm not showing this to you here, but if you take individuals who have the gene and look at their frequency of 438-2 usage in individuals with the gene, they're not different from controls. So that's an allelic difference as opposed to a frequency of usage difference in T1D versus control. But other genes, you can see a few of them are also underrepresented, but it's very subtle, as you can see from the scale here. And this is not surprising because we're looking in the overall repertoire. So if we saw something here, it would really be amazing if we saw it, but we don't. So then we said, okay, what about somatic hypermutation? The spleen is a very nice compartment to look at somatic hypermutation because it generally has a much higher frequency of memory B cells. But you can see here again, if you look at just the proportion of clones that have a mutation frequency of 2% or higher in the V gene sequence, and each dot here is a donor. If you look at autoantibody positive, which includes the T1D donors, versus healthy controls, you do not see a significant difference. If you look furthermore at the frequency distribution of SHM, you can essentially overlay the curves. So that's nice, but again, we're looking now at a mixed population. So a more rigorous way to do this would be to look at sorted memory B cells and ask whether their SHM frequencies differ. So we did that, pulling out naive IgA, IgG, and switch memory B cells from controls and from autoantibody or T1D donors. And if you now look at the somatic hypermutation profiles and you stratify by tissue, you get a rather interesting result. So you can see that in the switch memory cells, gosh, it's so hard to see this. Okay, so the memory cells in the spleen and the memory cells in mesenteric lymph node look fairly similar in terms of their somatic hypermutation distributions, but the pancreatic lymph node looks different. It actually looks like the SHM frequency is a little bit lower. And for those who look at memory B cells a lot, slightly lower SHM frequencies are reminiscent of certain kinds of activated B cells, such as ABCs potentially. So then we started to wonder, are some of these subset frequencies different in terms of B cells in the tissues? So in parallel, as part of the Human Pancreas Analysis Program, our lab has been generating a lot of flow cytometry data on these donors. So this is just an example of a gating scheme where we're pulling out by multi-dimensional flow different B cell subsets. Okay, so how do we look at these then? We analyze them, so you can see there's a lot of data here, all these different numbers of donors, and then the proportions of donors who have the different B cell subsets in terms of the different tissue distribution. So if you look at this plot here, you can see that really peripheral blood and spleen are the two main compartments where the subset distribution is different. The lymph nodes all look fairly similar in terms of their subset distribution. Now, how does this compare in T1D versus control? Okay, so this is a very busy slide, and the only reason I'm showing it to you is to say, okay, we've compared this in a whole bunch of different subsets, but the one to really focus in on is the one where you're seeing some, asterisks and stuff. So there may be some differences there. And so what subset is that? That's actually the class-switched age-associated B cells. And here I just made the graph a little bigger so those of us with older eyes can actually see it. So in peripheral blood, you don't see a huge difference, but if you look in spleen, actually there's a significant increase, and each dot here again is a donor. And you also see it in the pancreatic lymph node itself, in both the tail, and also we'll show you some more about the distribution. So what are age-associated B cells? This is a memory population that is characterized by the expression of the transcription factor TBET. It has a myeloid marker, CD11C. It can also express CD11B. Classically lacks CD21, 23, so it's really marking as a memory B cell. It's mostly found in the spleen, but also obviously can circulate. It increases with age, hence the name, but it also has this interesting property that you can see increased frequencies of these cells during flares of autoimmune diseases such as lupus in the peripheral blood. These cells can differentiate from other cell populations, and that usually requires some form of innate signaling. So TLR7 and TLR9 signals in the context of Th1 cytokines can drive the differentiation of these cells, and they're a very fascinating sort of autoimmune-linked B cell subset. So we were kind of excited when we saw this, and we wondered whether, if you looked a little harder at the pancreas and the pancreatic lymph nodes, how did these cells map out? And so I'll remind you, the pancreas sort of straddles the gut over here. So this is the duodenum. Here's the pancreas. And the pancreas has a head, body, and tail. And on the tail side is where you see the spleen. Okay, and if you look at the proportion of these class-switched ABCs, they really increase in frequency. So the tail has the highest proportion, and followed by the body and then the head. And it's really in the tail where you see the divergence, and the tail is the part that's closest to the spleen. So this is a very interesting kind of anatomic difference in distribution. So we wondered whether the ABCs were different in some way in T1D, not just in frequency, but in their characteristics. So we did a single cell analysis to look at this. And so here we took 6 different T1D donors that either had T1D or autoantibody or both, and some control donors. We sorted specifically on plasmablasts, ABCs, and resting memory, and then did the usual 10x genomics 5' end kit, looking both at RNA as well as immune receptor repertoires with paired heavy light chains. And a whole bunch of this kind of stuff, right, where you're trying to optimize the processing of data, which I'm going to happily skip. And then what did we find? Well, so if you look first off at just sort of basic cluster analysis, and we also looked at this in terms of differentially expressed genes. How can it be 1 minute? Okay, so we've got ABCs, plasmablast resting memory. You can see that these subsets, that they cluster nicely, which is great, and a good sort of encouraging thing of our sort, right? But then you can also arbitrarily cluster them into smaller clusters. Broadly speaking, the clusters don't separate massively, but you can do some differentially expressed gene analysis, and what the differentially expressed gene analysis shows is that we have more myeloid and inflammatory cell markers in the T1D autoantibody positive ABCs compared to controls. So then we wondered whether these ABCs actually span different tissues in some way, because the thing that was confusing to us about this is that ABCs are primarily in the spleen, but how do we get to ABCs in the pancreatic lymph node or the mesenteric lymph node? So if you look at clonal overlap and so all these red clones here are in the spleen that are ABCs. There's a significant number that overlap between pancreatic lymph node, but now they can't possibly be ABCs anymore because the frequency of those ABCs is very low, right? So we wondered then, if that's true, what's happening to the clone? Are they changing into another cell type in those other tissues? And so there's some evidence for this in mice from Nickerson and Schlomchik, in a mouse model where they're tracing ABC lineages and looking at them in different cell types, and we essentially see the same thing. So ABC clones can be found in different subsets of lymph nodes, and in different B cell subsets in the lymph nodes. So here we're looking just formally in a couple individual clones, and you can see in the yellow circles that we have switched ABCs and IgM ABCs in the spleen in yellow, right? But then we have IgG cells, IgG memory cells, that are sitting in the pancreatic lymph node. So these are very nice sort of demonstration of clones that can occupy different B cell subsets in different tissues. And of course, we're very interested in knowing how these clones evolve over time. So let's see, I have 10:51. The talk's supposed to go till 11, right? Am I wrong about that? Okay, so the signs that you guys have been holding up about 1 minute left are not true? Okay, we're good? All right, good, all right, phew. All right, so our next question is really, what do these cells bind? Are they actually autoreactive or not? And that is a hard question, as you can imagine. So you could approach this in an agnostic way and try to clone the receptors and then screen them in some heroic way. Okay. And then you can select clones. The alternative is to select them based on their size, their somatic hypermutation, their isotype, their subset representation, or, since that's very hard to do, isolate known autoantigen-binding clones and see if they overlap with some of these ABCs, right? So we're trying to get the top-down and bottom-up approaches to link somehow. So how do we do this? Well, so one way is to take the spleen again, because it's a rich source of B cells, sort on the IA2 binding B cells, do bulk gDNA sequencing to get deep clonal reads, but also complement that with single-cell analysis to pull out your antibodies. Why are we interested in IA2? So IA2 is one of the 4 classical T1D-associated autoantigens, but it's a receptor protein tyrosine kinase that actually is enzymatically inactive, but is involved in the transport or in the regulation of insulin exocytosis. The reason we think it's a useful antigen is that it has greater specificity for T1D disease than, say, GAD65. It's more prevalent in younger T1D patients. It's not confounded by insulin therapy, which is really important because insulin antibodies are made by virtually everyone with T1D when they go on to exogenous insulin. And it's a far more frequently found autoantibody than zinc transporter 8. And zinc transporter 8 also has the flaw that it is this horrible transmembrane, like 6 membrane spanning domains, and it's very difficult to use as a probe. It tends to aggregate. Okay, and then we have crystal structure data available, which is another huge advantage. So we pull these out by flow, we sort on the diagonal. We have since been improving this further with lots of additional bells and whistles. We pull this out of multiple donors. Then we subject those antigen-enriched populations to bulk sequencing of the antibody heavy chain, and then we analyze the V gene usage. So the first thing that's really nice as a proof of concept is that we have nice enrichment among the IA2 binding cells from multiple donors for VH3-7. So this was very reassuring to us. You can see this here if you just look at global repertoires that are not enriched for IA2 versus individual people where you do the IA2 enrichment, and each dot here is a donor. You can see there's a clear increase in frequency. Okay, so what do these cells look like if you look at them at single cell level? This is still very early days, but if you compare a few donors who are T1D or autoantibody positive to controls, you can see that the cell populations look a little different, but the numbers are also way different. So I take that with a huge grain of salt. But what is nice is that you can see some differences in terms of these single-cell RNA-seq B-cell clusters. So they have different HLA transcripts and stress response genes. So you've got these HLA transcripts, but that's not super surprising because HLA molecules are associated with T1D. But some of these other stress gene responses are interesting. So then finally, we pull out monoclonals. And so we did this again with our combined bottom-up, top-down approach. So we enriched for IA2 binding, and then we picked clones that we thought would be interesting based on their anatomic locations. Okay, now we're getting close. So out of those monoclonals, 3 out of 10 bound to insulin. And so you've got intracellular in gray, that's the original antigen we used as our bait. And orange is extracellular, and the blue is the whole full length. So these monoclonals are a nice sort of initial proof of concept. So now we're doing structural modeling and doing single cell analysis of many more clones and trying to use the modeling to predict the best types of IA2 binders. We have, more modeling that I'm not going to go into. So where do we go with all of this? So we're going fishing. The blood is like an ocean with many different fish. And what I hope I've shown you today is that we have some candidate bait to pull out fish of interest, right? So maybe some of these V genes, some of the age-associated B cells, there's plasticity in the subsets, there's some of these myeloid inflammatory transcripts, and of course the IA2 binding antibodies and modeling. And then we can use that for further repertoire motif discovery. And I'm going to skip this and just comment on the AIR community important thing, which is that the community is already using some of these HipApp data, but very eager to talk to any of you doing AI and machine learning to try to develop further a T1D-associated disease fingerprint. And then, of course, ultimately extend the analysis to motifs in the peripheral blood. Right, with that, thank you very much.
All right, so you have questions. Look at there.
We have questions.
And I do have a question, and that is, are age-associated B cells good at presenting antigen?
Yes, so that's a very interesting question. We don't know enough yet about the function of these cells in that context. What they are very good at is developing into antibody-secreting cells. So they seem to be transcriptionally and epigenetically poised after they receive TLR7 and TLR9 stimulation to become antibody-secreting cells. And they give rise to a lot of protective antibody responses in antiviral immunity, such as flu.
Wonderful. Thank you. Okay, so you gave the age of the patients, but do you know how long the patients have had T1D?
Oh, yeah.
Have you seen any impact on their repertoire as a function of duration?
Yeah, fantastic question. An anonymous asker, or no, not anonymous. Thank you, Paul. All right. So that is a great question. In these organ donors, it's a little bit challenging. For some of them, we do know about the length of disease. For others, we do not. It is a great idea to do this for the, but we don't have an absolute way to do it. One thing is for the antibody-positive individuals, it's actually very interesting there, I think, to compare single autoantibody positive to double or more autoantibody positive. Because with single, we really don't know yet whether that person actually is truly on their way to developing T1D. But with the double, we have high confidence. Mm-hmm. So that's also kind of like a window of, how long has the autoimmune process been going on. But it's not perfect, and I think that's part of why if we did this with repertoire analysis and we got a nice fingerprint, we could probably figure some of this out, right? Because we could figure out whether it's not just at the level of a single secreted autoantibody specificity, it's actually if you can get a whole slew of clones or fingerprints of clones that can see T1D autoantigens, then you could ask, how diverse are those clones? And maybe that gives you almost like a clock of disease development. But that's a fantasy right now.
All right, so let's see. Right, so when balancing classes and samples, what was the rationale to downsample samples to 20,000 clones per sample instead of, say, UMIs or read counts?
So we've done this a number of different ways. First, it's important to emphasize that in those initial experiments, those were bulk gDNA-based sequencing libraries, and so the UMIs for gDNA are very challenging to use and generally not super useful. So the downsampling was really to control for when you're looking in range map plots of clone size distributions, if you do not control for the denominator, those range maps will just show you essentially the clone counts, right? And since the clone counts were dependent on our level of sampling and not necessarily biology, we felt that it was important to make the clone counts the same in everybody and then look at the distribution of sizes.
Right.
I hope that makes sense.
That makes sense. Yeah. Have you looked at positive antigen specificity convergence between BCR and TCR in terms of specificity?
Yeah, that's a fantastic question, and we do have some parallel data sets that we're generating in the Human Pancreas Analysis Program, so whoever's asking that, come talk to me. It's a great idea, and
TBA,
yes, we're super interested.
All right, okay. In some T-cell mediated diseases, B-cell targeted therapy has been tried but did not work. Do you believe these were rather the wrong therapies here?
I'm not sure what that means exactly. I think in the case of the Peskovitz study, where B-cell depletion therapy was used in T1D individuals who had not yet developed full-blown diabetes, but were on their way, it clearly, the B-cell depletion clearly expanded or extended the honeymoon period. But B-cell depletion was not maintained, and eventually, in all of the individuals in that study progressed to type 1 diabetes. In a disease like multiple sclerosis, B-cell depletion therapy is being provided chronically for years. And so it could be envisioned that that might happen in T1D also, if it works, right? And I don't think that's really been tested yet. So in multiple sclerosis, an open question, of course, is when do you discontinue the therapy, right? So you just keep somebody on lifelong B-cell immunosuppression.
Yeah, can you guys leave me alone just for a minute?
Yeah.
Okay, so this is the last question because we have to then go away.
Yep.
And that is there any idea on the role/defect of regulatory B cells in type 1 diabetes?
Yeah, that's a great question. We do not know much of anything about it yet.
Okay, and then Brian Corey wants to plug something, and so you guys can read that.
Oh, right, but actually, can I just plug something for AIRRC7? So that was on the last slide. So we have a lot of our T cell data on AIRRC7. That's what, if we go back.
Okay. We can't do that right now.
There
I'm
it
sorry.
is. No, just, that's it.
Okay.
Okay.
All right. Okay. Nina, thank you. That was a wonderful talk. Really good. Thank you so much.